welcome to Botanic U. So today's topic is bacterial recombination and the plasmids. So first of all, what the recombination is? The recombination is the process in which a new recombinant chromosome having a different genotype from both of their parents. So recombination occurred by the combining the genetic material of the two organisms, and the primary focus in the recombination is the formation of the new genotype and the occurrence of the variation. So when we talk about the microbial recombination or the bacterial recombination, so there are the various types by which microorganism did its recombination. So most common type is the general recombination, in which is occur between the homologous chromosomes. Right, the exchange of the genetic material occur only in the homologous chromosomes and having the similar sequences as in the diploid organisms, and this is occur carried out by the product of the rec genes such as the rec A protein, and which is very important for the DNA repair. The general recombination is usually observed of two types, that is the reciprocal general recombination and the non-reciprocal general recombination. In reciprocal general recombination, the exchange is of two way, like a donor as well as recipient exchange their genetic material, and whereas in non-reciprocal general recombination, the transfer of the genetic material is only one way. Donor transfers the genetic material to recipient, but he itself remain unchanged. Means there is no change in the genetic material of donor occur. It only transfers the genetic material to the recipient. And the second type is the site-specific recombination. In this, the genetic material is not in homologous. This type of recombination occur in non-homologous type of the chromosome, and here the enzyme which is responsible for all this event of the recombination is specific for both the donor and the recipient, or we can say the virus and its host. Now come to the third type, which is usually used by the bacteria, are the replicative recombination. To recover, the replicative recombination accompanies the replication of genetic material, and it does not depend upon the sequence homology. It is used by genetic material that move about the chromosome. Okay, so here a complete set of the genetic material is formed. Very common example, it's transposable elements. Transposable elements carry out the replicative recombination. So here is the all recombination were represented pictorially. First one is in homologous recombination. The A plus, B plus, and the C are the homologous chromosomes, and the reciprocal recombination, here we can say this is the blue segments, which is a part of A positive, whereas in red segment is a part of A negative, and both the genetic material exchange. So this kind is reciprocal recombination. Whereas in second type non-reciprocal recombination, you can see that B positive was exchanged by a B positive fragment, but neither of the segment was transferred from the red one to blue one. So here the transfer is only one way, and it is called as a non-reciprocal recombination. Second one is a non-homologous recombination, which is occur between the non-homologous chromosomes. Here is A, B, C is a gene present on a one chromosome, whereas DEF is present on this other chromosome. So here both two chromosomes one chromosome ABC and the second is a DEF. They all are completely non-homologous, but the exchange in the segment of these chromosomes occurred. So the genetic material in which the segments exchange between the non-homologous pair, they are called as a non-homologous recombination. Next one, the site specific recombination. In site specific also the recombination occur between the non-homologous pair, but here the enzyme plays a very important role, which is specific for both the donor and the recipient. Now come to the replicative recombination. So replicative recombination as usual, the replication occurred here. A complete set of the genetic material forms its multiple copies, and it is not dependent on the sequence homology. And the very best example is the transposable elements, which follows the replicative recombination. So these are the terminology: X-genote, endogenote, and the merozygote. So X-genote, we called X-genote as a donor genetic material, or the donor genetic sequences are known as an exogenote, whereas endogenote is the genetic material of the receptor, and merozygote is a temporarily diploid DNA sequences. Recombination, during the recombination, the host genetic material, and next one is the merozygote. So during the recombination of the host genetic material, the recipient cell become temporarily diploid, and for a portion of the genome is called as merozygote, right? So during the complete recombination, here you can see that this one is an exozygote. This is an DNA fragments. It is an A, B, C, D, E, F. These all are the DNA fragments of the donor cell, and here is the recipient cell, which is ready for the recombination. And this one, here the recombination occur, but the fragment of the donor is not merged with the recipient. Here the ligation doesn't occur, and for that time period the cell is temporarily diploid, and this condition is known as the merozygote. And this one is a complete transformed cell. The transformed cell means that when the donor, the DNA fragment of donor is incorporated or inserted in the DNA fragment of the receptor, then it is called a transformed cell. Now come to the type of the recombination, bacterial recombination. The earlier one is also a type of bacterial recombination, but this is on the basis of the transfer. How the transfer of the genetic material occurs. So these are the three ways by which the transfer of the genetic material occur in the microorganism. So first one is the transformation, in which the DNA fragment, we can say the exogenote or the donor DNA fragment, is present naked in the environment, and it was taken up by the recipient bacteria. And we can say that the naked DNA fragment that was present in the environment, it might be because of the death of the some organism. Now come to the next one, that is a transduction. In transduction, the transport of the bacterial DNA is by the bacteriophage. Here you can say that here is in bacteriophage, which transfer is its genetic material into the bacteria. So the transfer with the help of the bacteriophage is known as the transduction. Okay, after the transfer of the genetic material, this genetic material was incorporated into the bacterial genetic material, and it completes all its process of replication, transcription, translation, and multiply itself inside the bacterial cell. And when it was completely filled with the virus genetic material, then it bursts out and the various bacteriophages forms. And third one is a conjugation, in which the direct physical contact is required, but the physical contact is only temporary, not permanent. During the contact a structure, a extended structure formed from the bacteria, which is called in pilus, which acts as a site of attachment for both the bacteria, and from which they exchange their genetic material. So here you can see that this is a pilus, a genetic material by which they come in contact and transfer their genetic material. So when we got to know, when we have to differentiate between whether the transfer of the genetic material was occurred by transaction, transformation, or the conjugation, the very basic criteria, the two very basic criteria on which we can differentiate is the cell contact and the sensitivity to DNAs. Okay, so when we talk about the transformation, no cell contact is required because it all is done by the bacteriophage, and here the genetic material is sensitive to DNAs because DNA is an enzyme which we can say damage the genetic material, that is the DNA RNA. And in case of transformation, the genetic material is naked, it is present in the environment. So if we add the DNAs over there, the genetic material is completely damaged and the transformation process is affected. So by this we can separate the transformation. In case of transaction, there is no cell contact required and no sensitivity to DNAs. But in case of conjugation, there is a physical contact, but DNA's sensitivity is absent because the genetic material transferred from one bacterial body to another bacterial body, and the genetic material never exposed to the environment, to the external media during the conjugation. So now come to the plasmids. So plasmids are an extra chromosomal small DNA molecule, okay, which have their own origin of replication. It is completely independent on the bacterial genetic material. Plasmid consists of the very few genes which is not necessary for the survival of the bacteria. Even in some cases it has been seen that plasmids can also be eliminated. The process by which plasmids are eliminated from the bacteria is known as curing. The commonly used curing treatments are acridine mutagens, UV and ionizing radiation, thymine starvation, and high temperature. So from this all these processes we can eliminate the plasmid from the bacteria and it doesn't affect the survival of the bacteria. So the very common types of the plasmids are the F plasmid, R plasmid, and the Cole plasmid. When we talk about the F plasmids, the F plasmids consist of the fertility factor and the gene which is essential for the pilus formation, and the complete pilus formation is dependent on these F plasmid gene, and it plays very important role in conjugation. And this is the general organization of the F plasmid, where this TRA gene, that is we can say TRA gene, is involved in pilus synthesis, or we can say pilus formation. It has their own origin of replication. Now come to the R plasmids. The R plasmids are the plasmids carrying the genes for the resistance to various antibiotics and antibacterial drugs. Most R plasmid consist of the two components, the resistance factor RTF and one or more R determinant factor. RTF encodes for the genetic information essential to transfer of the plasmids, whereas an R determinant are the genes conferring the resistance to antibiotics. So this is a structure, and this portion of the gene is responsible for the resistance factor. This plasmid consists of the multiple resistance factors like sulfonamide resistance, streptomycin resistance, chloramphenicol resistance, fusidic resistance, tetracycline resistance. So if we add all these antibiotics or antibacterial drugs in a nutrient medium, it can't affect the bacteria because they have the resistant factor against this gene. Now come to the last type of the plasmid. This is the Cole plasmids. So plasmids that code for one or more proteins, which is highly toxic to the bacterial strains, and that don't let harbor the same plasmids. Means that these type of plasmids secrete some protein which is only favorable for the particular strain of the bacteria. It can't let survive other bacteria in a particular environment. Like you have seen some grasses like that. If some grass has grown in a particular area, it can't let the other species or other varieties to grow in a particular area. So same thing was done by coal plasmid. It secretes some toxin material which is favorable for the survival of one strain but is unfavorable for other strains. So one of the example is the colicin protein which kills the sensitive E. coli cells. So this is an example. CMI encoding coal even like plasmid is a very common type of the coal plasmid. So that's all for today. Thank you so much for watching the video, and if you like the content then like, share and subscribe it, and also comment in the comment section that which content do you want the next. Thank you.